So let me just start by thanking the organizers, Pauline, Boris, Raphael, Stanislas Lyonnais, and say how much I appreciate this opportunity. You know, Imagine is, I think, one of the most amazing organizations. It's amazing to see its growth. And I thank the supporters of Imagine and Imagine itself and the organizers of this meeting for giving me the opportunity to talk a little bit. And, the title is Trials in Rare Disorders, but I've added Trials and Tribulations, which is an expression in English where the trials here means suffering. And the reason I put that in there is because while to this group, the idea of doing trials in rare disorders associated with autism is not so novel, to the greater world, it seems to be there's quite a bit of resistance. And I'll kind of talk conceptually first before giving some of the work we're doing. So, conceptually, we all know that there are rare disorders that are associated with autism. This is Rett syndrome. Just a couple of weeks ago, Huda Zoghbi and Adrian Bird received the Brain Prize for their work. And if you can see my mouse, I don't know. So, Huda Zoghbi identified, first of all, there was a definition of a syndrome, right? And then Huda identified the gene, and Adrian Bird made the first rodent model. Adrian then showed that if you reverse the phenotype, if you reverse the mutation in the mouse, you get a recovery of function in some ways. And all of this has led to, a robust series of clinical trials in Rett syndrome, right? So this is really a paradigmatic example of an autism gene and clinical trials that are going on. I would love to say we have some breakthrough successes. We don't. But I do think that with all the work that's going on now, we will start seeing some successes. But this is Rett syndrome where, the syndrome is well defined. And this is a placeholder that I'm going to come back to in the end. And this is about the trials and tribulations. There's a syndrome, which I'll talk about at the very end, called Mizuyoff syndrome. And this is what I can tell you about it now. The cause is well known. Nobody doubts the cause. You can test for that cause. There's high conversion, or it's highly penetrant, if you will. There are good models, cell and animal models, good preclinical evidence, just like with Rett syndrome, where if you reverse the gene deficit in a mouse, things will get better. The prevalence is such that it's, there could be a business plan, to use an American term. Lots of family foundations are involved. There are expert sites doing a lot of clinical and basic science research. The FDA or the EMA are very supportive of these rare disorder interventions. And then if somebody found a drug, it might be also useful in other related disorders. So this syndrome, I'm going to come back to at the very end. And so going back to Rett syndrome, as I said, it was defined by its phenotype, right? So this is classic hard genetics, where you ascertain individuals with a shared phenotype, and then you map out the gene. But there is a broader universe of neurodevelopmental disorder syndromes, which I'm calling here nonspecific, and they tend to share, one or more of these kind of 7 dimensions, if you will. And here you can't really just say, okay, I'm going to take all the girls that I find that have this regressive, apparently typical development, and then a regressive process, with very specific kind of cardio changes, respiratory changes, midline hand wringing, all the things that make Rett syndrome. It's much harder here, right? And not only are there many syndromes like this, but for any of these syndromes, even if we know the cause, there's any possible combination of the 7 kind of things on the right? So this is actually the Electronic Journal of Combinatorics. This is a complete Venn diagram of 7 things and everything. So for example, in purple in the middle, that group has all 7 of these characteristics, while the red ones on the outside may have one or the other. So this is, I think this is actually probably pretty accurate for some of the syndromes that we know about for NDDs, that you can have the mutation and have, not only disparate phenotypes, but also a subset of these nonspecific, so to speak, manifestations. Okay. So going to autism, I think you all can appreciate that autism is highly heritable. Estimates of heritability are 60 to 80% or more, but most of that risk is through common variation or SNP variation. But there's a non-trivial amount of it that is through rare mutations, de novo mutations, rare inherited variation, or things that are, for example, like recessive disorders. And so in terms of interventions, thinking about clinical trials in rare disorders with autism, it's kind of, it's in this slice of the pie, which is a minor slice. It's a small part of autism. So, Pauline mentioned kind of the big data part of all of this. There's a group called the Autism Sequencing Consortium. It's a very large group. It has 50 contributing laboratories, hundreds of participants. And at the time, it was very unique. And I just wanted to give a shout out to the people who founded the ASC in 2009. These are the two founders. You may not recognize them. The gentleman sitting down has retired. The gentleman standing up has come out of retirement. This is in 2009 when they signed something called the American Reinvestment and Recovery Act. And as part of that, a small amount of money of the total amount went to the NIH for some novel approaches to major disorders. And one of the things that was funded by that so-called ARA grant was this, in retrospect, kind of humorous collaboration across 5 sites where each one of us sequenced something. Well, there are many collaborators here, actually, and you can see Catalina Betancourt, who's one of your locals. But 5 sites were responsible for generating sequence data from 200 trios. So nowadays we would chuckle with just 200 trios. But the important thing was, or each of us ground away and better for worse sequenced, our 50 or 40 trios, of which only 175 actually made it through QC. We saw that, compared to models that were derived very elegantly from primates, the rate of de novo mutations were higher than expected. So nonsense mutations, so de novo, so a new mutation that was nonsense, was twice as high as you'd expect. I will tell you that the p-value was not very impressive. It was 0.04. That's also with a one-tail test. And I think you all know that when you're using a one-tail test, it's because the two-tail test wasn't significant. So, this was just a marginal finding, to be quite clear. But in that same year, other groups, and particularly the folks working with the Simon Simplex collection and Stefan Sanders, who will be speaking a little bit later in the day, did a similar study, and altogether there were about 1,000 trios, and 9 genes were identified when you put this all together. And it became quite clear, and this was kind of the intellectual basis, if you will, of the ASC, that, if we estimated how many genes could be discovered this way, the estimate that we're mostly working with is around 1,000 such genes. That number hasn't changed that much. It could be 800, it could be 1,200, but plus or minus. So with 1,000 such genes, you need quite a few cases to reliably find these genes. And so my 45 genes that I had, families that I had, wasn't going to go anywhere in terms of contributing to this discovery. And so the ASC this is in 2010, and we all got together. And most importantly, we had the funding organizations. This is Alan Packer from Simons. This is Thomas Leonard from the NIH in the room. And basically the idea was, and only a few of us really liked the idea, but the idea was that maybe we could share our data before publication prospectively. Instead of me publishing my first 30 trios and then the next person, 5 years will go by, and then we put the data together, we said, why don't we put the data together without publishing first? And that was pretty radical at the time. But, when the NIH is sitting in the room and saying, this is how it should be going, this is what we want to see happen, this is what we're going to fund, it becomes very hard for people to say, I'm going to keep on going with my 50 samples. And I think one of the reasons people look pretty glum here is because they didn't all agree with the concept, but they couldn't say it vocally, given that the funders were in the room. And so the ASC, I think, has been fairly successful and has kind of 4 foundations: obviously financial support, a lot of work on novel statistical tools. So, exome sequencing, genome sequencing, rare variation, incorporating case controls and de novo and inherited variation, all of that took methodological development. And we're very fortunate that a lot of the resources for the ASC go to methods development. I can't see all of my screen, but this idea of prospective data sharing and DNA sample sharing. So if somebody had DNA, instead of applying for a grant to get it sequenced, they would give it to the ASC and be one of, unfortunately, many hundreds of authors. And then, of course, the continued improvement in methodology. Okay, whoops, yeah. And the first paper came out in 2014. You can see now that it's a consortium paper because, when the second line of the paper says a list of authors can be found at the end, there are lots of authors. That's the negative about these big consortia. And then I'll briefly go through ASC, the most recent paper that came out last year. And I will say that this paper is already quite obsolete. There's now work by a subgroup within the ASC with twice as many samples. These are some of the leaders of the ASC. There's Catalina, one of your locals, and many of the people who actually did all the work who were more junior at the time, although many of them have their own labs now. And this is kind of the splash page from Cell. And the short story, which I won't go into in great detail, is that there were 102 genes that met our criteria. This is what it looks like. This is kind of a rare variant Manhattan plot. So basically, this gene has a p-value 10 to the minus 16 because there are multiple mutations, rare mutations in this gene in our cohort, and you don't see things like that in controls. So a low p-value or things towards the top have probably a couple of characteristics. They're not vanishingly rare mutations. It's not one out of all the ASC. There are multiple mutations in the ASC. You don't see mutations in controls, and effectively they're highly penetrant, right? So all of the genes here, right? I mean, anything, in fact, here could be considered for a clinical trial for a rare variant, right? And even the ones down here could be too. But of course, maybe this gene over here, there are fewer cases, and maybe harder to make a kind of a case for it, right? If you're thinking about it from a financial perspective. And I'll talk about a couple of these genes in depth in a moment, but some of the other things in this paper are important to kind of point out, and they have to do with this idea of precision medicine and autism. So firstly, and this is already well known, the genes that we find in autism don't fall into one category, right? So these are genes that are expressed early in development, and they are involved in regulating the expression of other genes. So they have multiple downstream targets. These are genes which are synaptic genes, right? I think when we first began to study autism genetics, we assumed all of our genes would be synaptic. Well, they're not. In fact, the majority are not synaptic, and they're involved in general developmental processes. So the idea, and this is my point, the idea that we're going to find like a magic drug that works for all autism is unlikely because the biology is so different. And one of the hypotheses that was, after that first couple of papers from other groups as well came out, the hypothesis was that maybe these genes are having their impact by modulating these genes. And so we could test that in the ASC. So the green genes over here in this circle, can you see the pointer? Probably maybe just nod if you can see it, because I'm relying on it heavily. Pauline, do you see this? Yes. Okay, it's not there. So these are the synaptic genes. These are many of the genes that regulate other genes. This circle over here are these regulatory genes with known targets, and these are regulatory genes that we don't know the target yet. And what you can see is there's a very, very strong evidence that these genes over here with known targets regulate those other ASD genes that are also involved in gene regulation, but there's no evidence that these genes with known targets regulate the synaptic genes. So even the simple hypothesis that these genes are having their effect through working through synaptic genes is also wrong. So these are truly independent. And that's why we chose this title, developmental and functional, which was, I have to say, ridiculed in the Twitter sphere. People thought that was the world's most ridiculous title because, of course, it's developmental and functional. But we chose those words because we consider these developmental genes and these functional genes, functional synaptic, so to speak. Another thing that was clear using single cell data was that you couldn't even say what kind. Not only was there no single cell type that was associated with autism, this is the excitatory synaptic, this is the excitatory neurons, this is inhibitory neurons over development. And what you can see is that the red just means lots of autism genes during this developmental process. So as excitatory neurons develop, you see more and more evidence for autism genes being expressed there. And the same is true for inhibitory, as well as OPCs. So it's autism is not excitatory only. It's not just neurons. It's very complex, right? One of the things that we know about these genes, all the dose-sensitive genes, and we knew it for many years, is that there's also a U-shaped curve, right? Too much is not good. So this was well known from copy number variation, where you can have a recurrent copy number variation, where you can have a deletion or a reciprocal duplication. And 22q11, one of the most famous ones, the deletion has a phenotype, the duplication has a phenotype, and it's true for virtually everything. This is one of the most significant papers in autism gene discovery of its time. And as you can see, for example, here is an individual with trisomy, with a balanced translocation. Okay, so dad has no phenotype. It's a balanced translocation. But as a result, there's a child who has monosomy, a child with disomy, and a child with trisomy of the terminal arm of chromosome 22. And what you can see is both the monosomy and the disomy have a phenotype, less severe here, right? So this is maybe Asperger's and other behavioral things, and this is severe autism. And so it's often the case that the duplication is somewhat less severe. But this idea of U-shaped curve, that dose is important, is very critical to thinking about therapeutics. They tend to be severe. All the genes we can talk about at the top of that sample are very severe. This is some of our work in Shank III, the same gene I just showed you here that Christelle, Catalina, Marianne, and Thomas discovered. And so you look at, for example, the IQ of individuals we ascertain with deletions, 50 some odd % have profound intellectual disability, and most have autism. And this is point mutations, and it looks a little bit better, but not much better. What's positive about the findings in the ASC is that most of what we're finding, and it's a circular argument because we're also we have the most power to find these things. Most of what we're finding is loss of function. So if you plot for any gene that we find, are there protein truncating variants or are they missense variants or both? You can see that most genes have lots and lots of protein truncating variants, and they're probably functioning through a loss of function mechanism. There are some examples where most of the mutations or all of the mutations are missense, and those are probably acting through a gain of function mechanism. And in fact, we know from like DEF1, KCNQ3, and so on, we know, and SNC1A certainly, we know their gain of function. But most of what we're finding operates through a loss of function mechanism, which means it's dominant, which I should have mentioned as well. It's a dominant disorder where one allele is perhaps dead. So you have a healthy allele that you might be able to target through gene therapy or any kind of gene-targeted therapeutics. So there's a way we can think about all the discoveries in autism, intellectual disability, as being an opportunity for a platform approach. Like how can we develop methodology to upregulate a healthy allele in the case of a haploinsufficiency condition? And the other positive thing, and this is going back again to Shank III, is that, and I've mentioned it also with Rett syndrome, studies, this is work of Gaoping Fang. There was a caveat in a follow-on paper, but then there's another paper that shows that most of what is true. Gaoping showed very elegantly that when you turn the gene back on, if Shank III is, if you knock it out in a mouse and you turn it back on, at different times during development, even in adulthood, you get some recovery. It's not perfect, but and earlier appears to be better, but there are many examples of this. Rett syndrome gene, MECP2, FMR1, many genes that have been studied in this way, at least in a mouse. If you turn the gene back on, you get some recovery. So it's not, there is something to be done even, in kind of postnatal development. And the other positive thing we're finding is that, some of the top genes are what I call relatively common. And I took that word from a review that Kathleen and I wrote, common in quotes, right? It's not common by any definition you might have, but it's common enough to have two properties. One, it's rare enough to be considered an orphan disorder, which has a very specific implication in drug discovery and development for biotech and pharma. But it's common enough that that you can see how there could be. You can study the individuals. You can think about. You can do deep phenotyping of them. You can kind of take it forward. And it's not to say that even if it was ultra rare, you can't take it forward. We know the famous story in Batten disease of Tim Yu, where he treated a girl with a Batten disease mutation, who was at the time and almost probably still today, an N of 1. And the other thing, and I mentioned this before, this is from the ASC's earlier paper, we know that most of these genes that we're talking about are highly penetrant. Their odds, their effect sizes or the odds ratios can be, 80, 60, 40. They account for most of the phenotype, if not all of the phenotype in the individual. So if you could target this gene in that individual, you will have an opportunity for intervention. Okay, so this is kind of what I just said. There's not a single pathway, and that's why precision medicine seems to be a critical approach. Overwhelmingly dominant, so the second allele can be manipulated. They're primarily loss of function, and we know a lot about, which ones are loss of function, which ones may be gain of function. Unfortunately, they show a U-shaped curve. We have to be a little careful. They tend to be very severe. And as I said, multiple examples, if you turn the gene back on, things get better. And they're, quote, fairly common and highly penetrant. So really the question I have about therapeutics and autism is, with our current state of knowledge, is there any viable alternative, right? Is there any, can you treat autism as a homogeneous condition, even if the individual does not have a mutation? If we know that there are all these complicated ways to get to autism, how are you going to, how would we expect that if we took, 80 people, that we took 80% of the individuals with autism where we have not found a mutation yet, that we could treat them as a single entity? We know that too much GABA, too little GABA, too much glutamate, too little glutamate, neurons in the wrong place, all these things will lead to autism. And so my position is that unless we can subtype autism, either by genetics or by other things, we will actually not be successful in developing therapeutics. And I'd love to be wrong. But I think that, there's a lot of effort still, because people think autism is a homogeneous thing. It's unified and it's a huge kind of market. So pharma's thinking about autism, and I think that they should be thinking about tiny subtypes of autism. I'm not the only one. So what are we doing at the Seaver Center? We kind of have 4 tiers for our drug discovery and development. We choose our targets through the ASC or other things. We do a lot of work in human neurons in vitro. We make genetically modified animals, and we do a lot of work studying the individuals with the mutations. So Shank III, I've talked about it several times, is one of the top genes in the ASC. We study we put a lot of effort in ADMP, DDX3X, FOXP1, and Shank III. We are starting to also work on CHAMP2 and Shank II. You can see that we go for the low-hanging fruit, the genes that are the more common ones. And I'll talk a little bit about Shank III. And what's interesting about Shank III, or Phelan-McDermid syndrome, is, we knew there was something going on at the tail of chromosome 22 50 years ago. So this is a series of reports of ring 22. So ring 22, because we could detect it early on with low resolution karyotyping, and you lose genetic material at the point of the ring. And you can see all these individuals with ring 22 feeding motor abnormalities, late milestones, low IQ, language delay, agitation, and so on and so forth. So all the things that, and even things that, I'll come back to in a bit, but, this really looks like one of these non-specific syndromes. And in fact, it looks like Phelan-McDermid syndrome, which is what we now call that syndrome where you're missing the terminal end of chromosome 22, or you have a mutation in Shank III, right? And so very systematically over many years, people began to map this syndrome. These are deletions, better and better, higher resolution, and ultimately, individuals with the syndrome, it looked like there were just a few genes at the very end that accounted for much of the syndrome. Some very nice work, again, by some of the leaders in the field at the time, showed that there were recurrent breakpoints, Bonaglia et al., for example, and other groups in Shank III. And this was really, there was good reason to believe that Shank III was going to be the driver of Phelan-McDermid syndrome. And of course, this critical paper came out that kind of confirmed it, not because they had a translocation that was already known, not because they had a deletion that was already known, but because they had a point mutation. It was an N of 1, but the point mutation was kind of the nail in the coffin, so to speak, that says Shank III is the driver of the syndrome. And in this case, actually, there's germline mosaicism. And so both individuals carried the same de novo mutation. Okay, so I said we do a lot of work with stem cells. I won't go into this, but this is 7 different patient-derived stem cells looking at gene expression. I'll come back to it in a minute. Shank III is down in our stem cells, which is pretty good. I'd like to kind of see that. We've made 3 different rodent models of PMS and Shank III. This is a survey of many of the published. There are 13 rodent models. And for example, many of them show the same thing: increased repetitive behaviors, decreased motor capacity, decreases in social behavior. So there is some consistency across the models, even though different parts of the gene are different, and some of them are, in our early studies, we showed that LTP, electrophysiology, was abnormal, and we showed that IGF made things better. So this is the animal missing 1 copy of Shank III, and this is the animal missing 1 copy of Shank III, but treated with IGF, and this is the control. So you can see this kind of decay of LTP was reversed by treatment of IGF-1. This is the rat model, and here we showed, all published, so I won't go into it. We saw very clear social recognition memory deficits and some attention deficits, both of which were reversed by oxytocin. So here is heterozygote, doesn't remember it's conspecific. You put it back with a black or a gray rat. It met the black one yesterday. It doesn't remember it. You give it oxytocin, it remembers it, and it spends more time with the new rat. These are attentional deficits. And again, if you give oxytocin, not only do you get much better memory of the rat it met yesterday, but their performance by multiple measures in an attention task is improved. And as I said, we also bring in lots of individuals with the mutation. This is the first paper in 2013 with over 30 individuals with the deletion. And then we've had a follow-up paper where we looked at individuals with point mutations. This was a critical paper to really show that most of the phenotype really seems to map to the actual point mutations in the gene. It was certainly strongly suspected, but we over-ascertained point mutations that were just becoming more common then. And I don't want to mention her name too much, but Catalina, who I know her lab is at least listening, has been a driver in everything to do with PMS in all of these studies. And so I mentioned IGF-1 in the mice, and the reasons we chose IGF-1 we can talk about, but one of the reasons was we knew we could easily put it in kids. It was already well known to be, it's only used in kids, in fact, with primary IGF-1 deficiency. And we did a kind of a pilot trial of that same drug, and there was an improvement in 2 things: social withdrawal, but also RBS, repetitive behaviors. There has been a replication study, which we've not yet published. The social withdrawal didn't get better, but the RBS replicated. So, it's still a small study. IGF is very expensive, but we're still optimistic. I showed you the oxytocin in the rodent. This study has just been completed by Alex Kalebzon, who is our kind of clinical director and leads most of the clinical trials. And unfortunately, the trial is completed, but the results aren't very convincing, unfortunately. And then here's a third example. This is a company called AMO that developed a compound that they then tried in some of the available mouse models of PMS, and it seemed to work very nicely. Their target is epilepsy, and the AMO trial is going on. So these are examples of, clinical trials for these rare disorders, using either existing compounds or compounds that were developed around a certain hypothesis and then tested. I think where the field has to go, and I'm coming back to our stem cells now, is that we really have to use kind of the traditional methodology of drug discovery. So here is the example. If we could get stem cells to show a phenotype in vitro, we could do high throughput screening of small molecules. We can use it to test kind of gene-targeted based therapeutics like CRISPR-A or an antisense. Shank III, for example, has an antisense transcript. Maybe we can interfere with the antisense transcript so that the healthy allele is expressed further, right? So I think that the field, and certainly I'm not, again, I'm not the only one, there's a certain tranche of biotech that is now thinking about these targets as kind of traditional drug discovery and development targets, with the addition, of course, that we can, the newest thing is that we can actually do gene therapy, SMA being the classic example. So a lot of people are taking their stem cells from patients or engineered stem cells, and they're beginning to do screens or validating kind of gene-targeted approaches for these various disorders. And I think that's where we're going as well. I think I have a few more minutes. I wanted to talk about my newest favorite gene, DDX3X, which is one of the other ones we talk about. I'll say that, FOXP1, Pauline, and the CBER Center are already collaborating on that gene. But DDX3X is pretty new for us. And I thought might be worth discussing. ADMP, there's a very strong, multiple European groups working on ADMP. But DDX3X is a pretty recently described gene that accounts for 1 to 3% of intellectual disability and developmental delay in girls. So it's really quite striking how this is a syndrome we never knew about. It's also an autism gene. We see it in the ASC. This is work by Dan Geschwin and Dennis Wall and their cohort. And DDX3X is significantly associated with autism. This is a paper from the DDD, the Deciphering Developmental Disabilities. And the important thing to see here is that DDX, is really kind of off the charts in terms of the number of damaging mutations in females. It's certainly on par, if not higher, than Rett syndrome. We don't have good data on frequency really, but this is not a rare. You've heard about Rett syndrome, MECP2. DDX3X syndrome is probably around the same or even more common. And so, CBER Center just completed. Dorothy Grice is the PI on this. Just completed. And put online detailed phenotyping. So this is what we do when a family comes in. We do all of these things here. It takes 2.5 days. Plus we do biomarkers, EEG biomarkers, eye tracking biomarkers, other kinds of biomarkers in order to map the phenotype, because, none of these disorders have autism as the primary finding when you meet the families, right? They're much more concerned about, lack of sleep. I mean, there are many targets, if you will, that you might be able to for intervention. This is only 15 individuals because the pandemic kind of stopped things dead. But what you can see is, sensory abnormalities very clearly, low IQ, autism, and so on. What's interesting about this disorder, and this is what's in this color here, I don't know, beige, are point mutations that are PTVs, protein truncating, and these are missense mutations. And what you can see is, by many measures, the point mutation is less severe than the missense. And here is one of those cases where the point mutation is a loss of function, and the missense is probably a gain of function. Right? So that we can do genotype, phenotype within this. And this is saying the same thing, comparing them. And then we made a mouse, and Sylvia de Rubes is following that up. She's put her recent paper on bioRxiv. Here are some lamination deficits. Oh, I took out the paper. There's various behavioral deficits as well. Okay, so this is kind of what we're doing, and we're not the only ones, but what we're trying to do, which I think is unique, and I think probably Imagine is one of the few other places that's doing this as well, is we're trying to do more than just one thing. Some sites are great at phenotyping the patient. Some sites are great at having animal models. Some sites are great at having human neurons. But we think there's value to kind of having this whole thing because, we believe that if somebody came from biotech and said, hey, what are your top genes? And we'd show them the data from ASC. And then we'd say, why don't we work on this gene? And we'd say, listen, we already know the phenotypes that are worth going on. We're clinical trial ready. We have stem cells and we have animals. So, if you come up with if you do a high throughput screen with our stem cells, it could be tested in the animals. And if it works, you can do your magic medicinal chemistry and it could be tried in patients, right? So we think that the academic sites can do all of this, and biotech and pharma, or and of course, some academic sites and some hybrid sites can actually do high throughput screening and med chem. But most academic sites, tend to do this a little bit better. Because we haven't had a lot of people knocking on our door, we actually hired somebody from industry 2 years ago who's actually setting up for those genes, the high throughput screens, and to take it at least as far as we can, and then maybe get some interest from partners. Okay, this is that syndrome I talked to you about before. And I showed this slide to a group of pharma and biotech people. And I showed the slide, just as with you, I showed the slide at the beginning. And I said, if I showed you the syndrome, would you want to work on it? And they all said, absolutely. That's the perfect syndrome. And they all raised their hands and said, tell us more. Tell us more. And then I went through what I just did for you. I went through Shank III as an example, and the cause is clear. It's Shank III mutations, right? You can test for Shank III mutations. If you have a Shank III mutation, you're going to have a syndrome. The models, cell and animal models, have high construct validity. I showed you that if you turn the gene back on for Shank III, things get better. We have a pretty good estimate of its prevalence. Certainly, there are actually there are multiple family foundations all over the world that will love to partner with industry. There are places that are doing deep phenotyping. We know the phenotype. We know the endpoints. The FDA is very supportive of these rare disorders, EMA as well. And presumably, if there was a Shank III treatment, somebody could try it in another neuronal mutation associated with autism. So I went through all of my data and I showed them this slide again. And I said, Shank III, doesn't that meet these criteria? What about ADMP? What about FOXP1? What about all the genes at the top of the list? And they're like, well, yeah, but we're not so sure, and that's where it all falls apart, right? Show this list and people say, I definitely want to do that. Show the real data. They're kind of like, are you sure about this estimate of prevalence? What if you're wrong by a little bit? And so that's why after that meeting and a couple more like it, we said, we're just going to have to do it in academia. Take it a little further in academia. So there's no such thing called Mizuyoff syndrome. Mizuyoff is a Hebrew word for fake, but there are many in autism, in intellectual disability, in epilepsy, and developmental delay more broadly. There are many examples of genes that have all of those criteria, or most of them so far. And if they don't, it's only because we didn't test them yet, right? So there are multiple opportunities to get an entree into all these kind of non-specific disorders with a rare disorder clinical trials approach. And I think, and especially because, as I said, they share some properties of being kind of haploinsufficient, for example. You can even think about it as a platform approach. Instead of thinking about gene by gene, can we do something that would work with 5 genes or 10 genes at a time, right? So I think there's a way forward, and this is a call out to all the industry people to get behind what we're all doing here. So I want to thank you, and I'm sorry I think I went a little over, but happy to take questions, assuming we still have time. And thank you for your listening.